right? So we had 684 out of 2,700, right? We get 0.025 times 100 equals 25%. 25% of the offspring are recombinant, meaning neither one looks directly like the parent, so 75% are parental phenotypes. Do we understand everything we've done so far here? Cool. Does the recombinant frequency you calculated in the previous question support or reject your prediction as to whether these traits are supports the prediction that they are on the same chromosome because it's less than 50% recombinant. If it's less than 50% recombinant, remember this was in our lecture a couple days ago, less than 50% recombinant means it is probably on the same chromosome. So you just figure out the percentage of recombination. If it's less than 50%, same chromosome. If it's more than 50%, more than likely different chromosome or far apart from each other on the same chromosome. The frequencies can be used to map genes on chromosomes because frequency represents the relative distance between genes. The greater the distance, if you don't stop. The greater the distance, the greater the chance of crossing over event. Meaning, the greater the distance, the greater the probability that the genes will not be inherited together. So crossing over occurs during prophase 1. What happens during the crossing over event? The synapse occurs, right? There's the chiasma, everything goes. They stick to each other. When they get pulled apart, part of it sticks with this chromosome, part of it sticks with this chromosome, and we get a different looking chromosome than what we had. The gene mapping. When recombination frequencies are used to map genes, the result is known as a linkage map. A linkage map will show the relative locations. We don't know exactly where it's at on the chromosome just by gene mapping, like this, linkage mapping. We know where they are relative to another gene, but we don't know it's here, it's here, it's here, right? We just know relative to something else. The units used on linkage maps are known as map units. Map units are equivalent to the recombinant frequency. So a 5% recombinant frequency means 5 map units. 50% recombinant frequency means 50 map units, right? If you look at A, B, and C, and the distance between A and B is 5%, and B and C is 10%, and A and C would be 15%, right? Or something like that. That's how all this works. So examine the chart below showing 4 different genes, B, C, D, and E. Start by finding the genes that are furthest apart. C and E, 40 map units. So if we look, B and E is 25, C and E 40, C and D 35, B and D 20. So we put C and E furthest apart from each other. 40% means the recombinant is only 40% of the time, right? So they're furthest apart. Or I'm sorry, the linkage is only 40% of the time. To determine the location of the remaining genes, use trial and error. Think of the chart like a riddle and frequencies as clues. It may take some time. Once you have all the genes in place on the chromosome, use subtraction to finish filling in all the map units and genes. So we know that C to E is 40 map units. Right? And we know B to E is 25 map units. So you could put B somewhere, and then you go, okay, B to E, that's 25 map units. C to D is 35. So this C to E, C to D has to be less than C to E, but you can't figure that out until you figure out how much B and D are. Right? So if you look and you're like, oh, C and D are 35 units, right? And B and D, we don't ever get B and C, do we? We're never given the information for B and C. But we can figure out, right? Because this is 40 from C to E, and it's 25 from B to E, and we know this is 5, right? The 15 and 25. This is the different. This is the 15 map units here. Does this make sense or no? Does it? Okay. Do we understand the C to E? We find the ones that are furthest apart. Right? So on that table, that one makes sense. C to E, 40%. It's 40 units away from each other. And we know from there, we've got C to D is 35. So we've got C, D is 35. Herein lies the problem. So I've got this chromosome, and I know C and E is 40, right? And so most people, when they're like D is 35, they'll be like, oh, here's D, right? And this distance will be 35. But could it also be that you have D here, C here, E here, this is 40, and this is 35? Is that possible? Let me draw it bigger. So there's always at least two probabilities for the chromosome. We'll do this. So here's one, and here's two. We know, let's go, CE is 40%, right? What were the others? BE is 25. CD, 35. And BD, 20. So we look at the furthest, CE. Okay, if they are in fact the furthest from each other, that would put one on each end of the chromosome, right? And this would be the 40%. Yes, no? What if we just put C here, E here, and we're like, okay, this is 40%. And when we look at the CD next, C to D is 35. It doesn't tell you where it is in relation to E. It's only talking about CD. So one possibility is that D is here, and this is 35, which makes this 5, or D is here, and this is 35, which would make this 75. Right? It could be either one. Right now, we have no idea. Does that make sense? Cool? So this is why. So this is why, like, we talk about relativity and not exactly where everything is. So, okay, we got that. So we need information that is going to relate B and E and B and D. We know E is this far away. So let's work with E. B is at 25% from E, right? So if it's 25% from E, then that could put B here so that B to E is 25%, which would make C to B 10%. Or if we're going B to E, E is here, 25%, it's gonna be lower than this 40, right? It's 25%, so if B is here, and this is 25, well then this has to be 15, because this 25 and this 15 equals 40. Does that make sense? What do you mean this?
I'm grasping it.
Because we know C and E has to equal 40. And depending on where you put B, right, if, quite honestly, if this chromosome were longer, we could put B out here as long as this still equaled 25, correct? Because we don't have information about B and C. But we do know B and D. That's where it starts to put everything together. If B and D are 20%, and B and E are 25, that means B, D, and E are all fairly close together. Right? They all have a similar number. So that tells me that B, D, and E have to be close, or they're closer than they are to C. C's at 40. C has to be the furthest from all of them. So C is here, and so I can go in order, the 20, the 25, and the 35, right? If C to D is 35, and then B to D is 20, well then it has to be, if B to D is 20, it has to be closer than the C is. C is at 35. So that puts a difference of 10 between these two. It still has to be 35 units from C to D. It has to. Right? If this B is here, if I put it over here, it's 20 units away, B. And now I'm looking at B and E. Oh wait, that's, you've got to, let's take this one out. You've got to ask yourself, we know this. Now, what are the combinations then? It can be C, B, E, D, C, E, B, D. It could be C, D, E, B, or B, E, or it could be B, E, C, D, right? There's all these different combinations that it could possibly be on the gene. You need to use the mathematics of how many times they're recombined to give you those math units. Does that make any sense? No, huh?
I think because you added things and it kind of made it more confusing.
I added what?
I got lost at like when you said 25%. Like you added another 25%, right?
Added another 25%.
Because you said that if you make the 25% like in a different area, then it would mean the same thing.
Alright, let's do this again. B, E, C, E, C, D, B, B, and we're at 25, 40. Alright. So when I put this 25 in, that's what goofed you up? Because, oh, are you good with this? That C and E are the furthest apart than any other letters on there because they have the highest number. Cool? Next to that, we've got 35% C and D. So the next furthest apart is C and D. So that means D is either here or here. D's units are smaller than C and E's, right? But C and D is 35. This could be 35, or this could be 35. We know that this is 40. And quite honestly, could E be over here at 40? Absolutely. Either way though, D is shorter than that. Does that make sense? Is that okay for that?
Yeah.
Okay. Then we've got the 25% for B and E. So what that tells me is that the 25 could be here or here. As long as it's 25 from E, we don't know. Right on? So again, so this distance here is 25, or it's 25 that way. We still don't know. Does that make sense? Okay. Then we've got BD is 20. If this is 25, and we know D is in between C, D can't be on the other side of E, because C to D is smaller than C to E. That puts D here or here, right? So if B to D is 20, and B to E is 25, it can't, right? You can't be 25 this way and only 20 this way. If it were only 20 this way, that would put D here. This B to E tells you that D has to be here, and it moves the B to that side. Because it's impossible for B to E to be 25 and B to D to be 20 with D in here. It just mathematically doesn't work. Does that make more sense? And that's what you have to do, is kind of reason that out. So we know that this is impossible. We know B can't be over there. Has to be in here somewhere, so we can just go ahead and we'll put B here. And that makes this distance 25. Wait. Yeah. So B is here, and this distance is 25. We know this distance is 35, and this distance is 40. If this whole length from C to E is 40, and this is 25, and that's 35, right? We know these dudes, because this is at 35, and we know that's at 40, we know the distance that B is from D. Don't trip. That's what all this is. So D to E has to be 5 units, and that backs B up to here. Because if this is, this will be then 5 units, because this is 25 and 5, well, 10. Did I write it wrong? 20? Yeah, 25, 20, 35. Yeah. When you add up any of these guys that are in between, they have to add up to the total distance between C and E. Does that help or make sense? Am I confusing anybody more? Are we all good? Let's see if we can figure the next part and see if it helps or not. Yeah, gene mapping is not the simplest thing, but it's not so specific either. So if we make a linkage map on the chromosome for each data set given, we have MP, MJ, RJ, PR. First thing we're gonna do is look at what? What do we wanna look at? What do we, the ones that are furthest, right? So we've got a 42. So we know M and J are the furthest apart from each other. So we just put them on the ends. We still don't know where the others go yet. And we know that M to P is 21, R to J is 16, P to R is 5. So let's go to the next largest, which is the 16. So M to P has to be 21. It could be 21 this way, it could be 21 this way. But we know that P to R is 5. So if we put R over here, we can't put P over here. Because that would be 21 plus 42, that would be 56 away. So the P cannot go on this side of the M. Does that make sense? Because it has to be at the same time 21 away from M, but only 5 away from R. That puts it in between the M and the R. Yes, no? So we've got M, P, R, and then we look at the J. Well, we know, oh wait, we didn't know that was. So P to R is 5, R to J is 16, so we have to be here for 16, because we have 42 for the M to I, or M to J, my fault. If we went M to J and J to R was 16 and we put the R over here and P to R was 5 and M to P was 21, it doesn't fit. The math just doesn't put it there. That's all this really is okay, mathematically, where does something have to go? Let's do it. You get it? Got another one, 6 genes, and it's again the same thing. Let's find our largest, 48%, C and B, that puts them furthest apart. And we have a C there we can work with, a C there that we can work with. We don't have any other B information. So we've got to work with C. C to D is 5%, so it's either going to put the D here or here. A to E we don't need yet. D to E, if we have this D, right, the D to E is 25, C to E is 30. So if we do D to E over here, the C to E would put E in here, but then that doesn't work with the D to A and the A to E. It's just a whole, like they say, it's just a puzzle. Just go mathematically, and it's a logic puzzle. If this is this, then this has to be that. Put them in order, do the numbers add up. The distance, does the distance from here to here, which is 30, right? Is that equal the distance from here to here when you add these guys up? Well, A and E is 7 plus 18 is 25 plus 5 is 30. So this 30 is the same distance as this 30. That's it. That's all there is to this. Are we okay? Are we sure? Okay. Another one, this one was fairly straightforward. Non-Mendelian, complete, this wasn't homework, right? Was it? Yeah. Part of homework, it was. Complete, incomplete dominance, codominance. Complete just means if the dominant allele is there, whether you have 1 or 2, it's going to show up. Incomplete dominance means you're going to get a third trait that you didn't see in either of the parents, red plus white equals pink. Codominants, they're cooperating together, they're gonna coexist, so red and white will show up together. One gene at one locus affects a gene in another locus, that's epistasis. That's basic common knowledge. Why are there more X-linked disorders than Y-linked disorders? The Y chromosome is really small, it does not code for much. X chromosome, everybody has an X chromosome, not everybody has a Y chromosome. So the more X chromosomes there are, the more chances you will see a disorder on the X chromosome, right? Affects more males than females. So this, right, question 4, because males can't get an X chromosome from their father, mitochondrial, chloroplast, and DNA only inherited by offspring from the mother, because it's only in eggs. The sperm is too small to have it, I suppose, right? They are only located in the egg cells, so we can't ever get those from the father. Linked genes always be inherited together? No. Even if they're linked, they don't always have to be inherited together, because wherever the synapse occurs is going to be an exchange. We could break that linkage. It's not as common, but it could happen, right? This is why we don't see the 1/4, 1/4, 1/4, 1/4. And yellow coat is dominant to gray coat color. Inheriting 2 copies of the dominant yellow trait, however, is lethal. This is where we're saying, right, you don't see, in a cross between 2 yellow mice, what proportion of the offspring is expected to be yellow, what proportion is expected to be gray? So since 2 copies of the dominant allele is fatal, we wipe this dude out. That's not possible. So 2/3 yellow, 1/3 gray. Right, the homozygous dominant is lethal, so we have a 2-to-1 ratio. True breeding black feathered rooster mated with true breeding white feathered hen. They're all blue feathered. What does that tell you? That means that the blue and white give us a different trait, right? We're not getting black and white, we're getting something different. What is the simplest explanation? Incomplete dominance. Neither trait is shown. We get a 3rd trait. What would be the phenotypic ratios of the F2 generation? We get a 1 to 2 to 1 again. 1 black, 2 blue, 2 blue and 1 white. Just another little Punnett square. True breeding red snapdragons with true breeding white, we get pink. This is incomplete dominance again. Am I going too fast? Look like you were stressing. This is incomplete dominance again. If a self-cross, the F1 generation produces F2 generation, 600 offspring, how many are expected to be red? 1/4 would be red. 1/4 of 600 is 150. Simple, yes? Human blood groups, A, AB, and O. Oh, roan color of the cows, I forgot. Example of codominance, white cow mated with a red cow, we get the roan. Predict the phenotypic and genotypic ratios of the offspring of cross between a roan cow and a white cow. Here you have roan, here you have white. So roan, white, 1-to-1 ratio. 50% roan, 50% white. No red. Now blood types. So blood types are A, which can be IA or lowercase I, or 2 uppercases, B, AB, and O. Man who is type AB marries a woman who is type O. What are the chances their child will have blood type AB? Well, AB, if they get AB, there's problems, right? You can't. The O cannot pass on an A or a B, so that's 0% chance. If he's not type A or B, he can't have a lowercase I, which gives us the information for O. So it's impossible to get O because you have to have that. You have to have 50% type A, 50% type B. The type A's will have A and a lowercase I. The type B's will have IA and a lowercase I, or IB and a lowercase I, right? And that's what you see here. That's the only thing that can happen. We good there? Plants provide examples of epistasis, sure. Where multiple genes can control phenotype. In the case of corn, there's a gene that controls color. The kernels can either be blue or red. Different gene determines whether or not that color will actually appear in the kernel. The dominant allele inhibits coloring, meaning the kernels will be white. So if there's a dominant A present, we don't care what the rest of it says. That's what epistasis says. All colors, either red or blue. So if a plant is heterozygous for both traits, is self-crossed, what is the resulting phenotypic ratios of the F1 generation? Hint, this is a dihybrid cross, but since epistasis does not follow Mendelian genetics, it will not be a 9:3 to 3:1 ratio. If it is heterozygous for both traits, right here, it's heterozygous. This is the trait for color, this is the trait for epistasis. Right? So we make our regular Punnett square, or you can do this by the fork method. What is this going to be? What will this one be? So if we look, blue is dominant, red is recessive, what will this one be? If it's not epistatic, it would be what color? Blue. This is an epistatic gene, so we have to look at trait A. A dominant trait A means what? No color. Right? So as soon as you see this, I don't care if it's one or two of them, it's like, oh, white, 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 blue, blue, white, white, blue, red. Right? Look for that dominant epistatic gene, and then you don't have to worry about what the rest of it is. If that dominant allele is there, we just, I don't care. Nothing else matters. It's like Metallica. Nothing. Okay, so that song? Look it up. Great song. Okay. Another epistatic example with mice. Agouti is the wild type, yellow coat color, which is dominant to black. A different gene controls whether or not the pigment is deposited in the fur. Dominant allele shows the pigment, while the recessive allele causes albinism in the mice, regardless of the other allele. If a mouse with the genotype heterozygous is crossed with a mouse that has that genotype, what will the phenotypic ratios of the offspring be? Well, this can only contribute a recessive and a dominant, right? So that's it. This is homozygous or heterozygous for both traits, so we have to do all four alleles, and we get this gene combination: 2 agouti, 2 black, 1-to-2 or 1-to-1 ratio. Color blindness, X-linked disorder, normal vision, dominant to color blindness. A colorblind male as a child with a woman who is a carrier for the disorder was the probability. So he's colorblind, so he has to be XCY. She's normal but a carrier, XN, XC. So this person here, this female here, carrier. This female here, colorblind. This male here, normal. This male here, colorblind. 50% chance of a daughter being colorblind. 50% chance of a son being colorblind. Good? It's a monohybrid cross that we just look at the X chromosome. Duchenne muscular dystrophy, again, X chromosome. X-linked recessive disorder. If a man is normal, as a child with a woman who is a carrier, what is the chance the child will have the disease? So it is a recessive disorder. Normal, carrier, normal, having the disease. Sure. Eye color and Drosophila, which are fruit flies. Determined on the X chromosome, dominant version of the allele causes red eyes, recessive causes white eyes. Females can have red eyes or white eyes. Males only receive one copy of the X chromosome. So whatever allele version they receive dominates or determines their eye color. If a white-eyed male is crossed with a red-eyed heterozygous female, what would the phenotypes be? Well, if he's white-eyed, he has to be recessive, right? And if she's heterozygous, she's here. So here we have one of each. Here we have white, red, and white. 1-to-1 ratio, white to red. Not all. And it doesn't matter, male or female, it's still the 1-to-1 ratio. Are there any others? Do you need me to go through every one of these, or are we good? You need me to go through them all?
Yeah.
Yes?
Yeah.
Why?
I don't know, dude.
Do you not know how to do it? Which ones don't you know how to do it?
The box is kind of confusing.
Which box?
The, what's it called? The X, oh, yeah.
The Punnett square?
What's confusing? It's just like all the letters and stuff like that. It's like what they use.
Okay. They always give you the letters, or they give you the genotype in words. If you're told it's heterozygous, it's one of each letter, right? If they tell you this trait is dominant to this one, and you know the recessive is mixed, well, you know the recessive genotype, right? It's a matter of just turning heterozygous and homozygous into letters, and it doesn't matter what letters you use, right? It's just if it's heterozygous, it's one capital, one lowercase. If it's homozygous, it's two of the same. If it's homozygous recessive, if it's homozygous dominant, it's two capitals. Right, and that's it. It's just turn those words into little letters and then move on. Is this one? Alright. We'll look at this one because it's recombinant. Frequencies can be calculated adding numbers of offspring or different from the parents and divide by the total number of offspring. Let's say you study fruit flies, you cross a wild type gray female with vestigial black male. Gray, wild type, that's what we see, so that's dominant. Wild type, vestigial. Vestigial is where the placement of the wings are, right? With a black, with a vestigial black male. So wild type gray with vestigial black. F1 offspringers follows 650 wild type gray, so 650 look like the mom. 595 vestigial black, so look like the dad. 107 wild type black, doesn't look like either. 91 vestigial gray, doesn't look like either. What is the recombinant frequency between the genes for the wing type and body color? So you have to look at them all, get your total offspring, 1,443. We have the recombinants, 107 and 91 is 198, so 198 of the 1443 is 14%. So the recombinant frequency is 14%. Which means these genes are really close to each other on a chromosome. Are we almost done? Man, this time goes quick. We need one for 85. Then we did the notes on chi-square. We did this. All right, and then we had chi-square practice problems, right? Just 2 pages of that, right?
Yeah.
Okay. So here's what we'll do is I'll try and run through these. Do I have time right? No, I don't. We'll try and run through these tomorrow morning. Did you read the M&M lab? You know what you're doing? So I've got the M&Ms, so if we run through these quick enough tomorrow, then we can run the M&M lab.